Hello everyone, this is LATHA AKULA. In this video, we'll start the new concept, that is the plasmolysis. Plasmolysis. If we see a plant cell, the behavior of movement of water movement in the plant cell, that depends upon the surrounding solution. Here, there are three different types of solutions: hypotonic solution, hypertonic solution, and isotonic solution. If we see isotonic solution means what? Here, concentration of external solution is same like that of the cell cytoplasm. That means both are similar. There may be no differences between the cell cytoplasm and external solution. That means isotonic. Hypo means less. Hypotonic solution means what? When compared to cell cytoplasm, the external solution is less concentrated. That means it consists of less number of solute molecules. And third one is hypertonic solution. Hypertonic solutions means what? Here, the concentration of external solution is more concentrated than the cell cytoplasm. That means the external solution consists of more number of solute molecules when compared to the cell cytoplasm. It consists of more number of solute molecules and less solvent. Now we'll see what is plasmolysis. If a plant cell is placed in hypertonic solution. Hypertonic solution means what? Highly concentrated solution, right? Here what will happen? When the plant cell is placed in hypertonic solution, water moves out of the cell, out of the plant cell. That is because of the osmosis. In the osmosis, what we have studied, water moves from the region of higher concentration to the region of lower concentration, a semi-permeable membrane, right? Here also, the concentration of the solvent is more in the cell when compared to the concentration of the external solution. Then from inside the cell cytoplasm, the contents present, they starts moving out. This upward movement of the water that causes shrinking of the cell membrane. This phenomena is called as plasmolysis. Here, first, the contents present inside the cytoplasm will be removed outside. After that, the vacuole sap also removed. And here, the cell is said to be the plasmolyzed, and the phenomena is known as plasmolysis. And this phenomena is a reversible process. That means when plasmolyzed cell is placed in hypotonic solution. When it is placed in hypotonic solution, the plasmolyzed cell will be deplasmolyzed. That means it will get again the water. That means cell becomes turgid. Here in the hypotonic solution, what will happen? It is less concentrated, right? Then the water will move from outside to inside. In hypertonic, from inside to outside, the water will move. Whereas in hypotonic solution, water moves from the higher outside external environment to the inside the cell. So because of this, the cell becomes turgid. Turgid means what? It will swells up. Because of the entry of the excess amount of the water from the hypotonic solution into the cell, it will be turgid. So because of this turgidity, what will happen? The pressure build up inside the cell against the wall. That pressure is called as turgor pressure. For instance, if this plasmolyzed cell is placed in the isotonic solution, what will happen? Isotonic solution means what? Both the concentrations are similar, right? The cell will be flaccid. But if it is placed in the hypotonic solution, it will become the turgid. That means it will swells. Here, when the water diffuses into the cell, it builds some pressure against the wall. That pressure is called as turgor pressure. Turgor pressure exerted by the protoplast against the cell wall due to the entry of the water. That pressure is called as pressure potential. It is denoted by psi p. And here, if you see the plant cell walls, here, because the plant cell walls are highly rigid, right? So because of that rigidity, they are able to withstand this turgid condition, shrinking, and all this flaccid condition and all this. Unless what will happen? The cell walls, the cells maybe get ruptured. This turgor pressure of the plant cells that helpful in maintaining the erect condition. Next concept is imbibition. Imbibition means what? Imbibition is a special type of diffusion. Here, the water is absorbed by the solid. Water is absorbed by the solid. And not only that, because of the absorption of the water, the volume of that substance also increases. That process is called as imbibition. Examples for the imbibition is when we soak any seeds in the water. After some time, the size of the seeds increases and the seeds also absorb the water. That is one type of the imbibition also. And if we keep the dry wood in the water, the dry wood will absorb the water and its volume also increases. This is also one of the example of the imbibition. And this imbibition is a reversible process. When we keep them away from the water, they loses the water and they will come to the normal position. And the solid substance which is absorbing the water is called as imbibant, and the liquid absorbed is called as imbibate. Liquid absorbed is called as imbibate. And the rate of imbibition increases when the temperature increases. The rate of imbibition also increases. The plants are the good imbibants because they were hydrophilic in nature. Hydrophilic means what? Water loving. They like water. And examples for the plant imbibants are phycocolloids like agar. It is obtained from the red algae and it is a good, best imbibant. And not only that, there are some other imbibants like proteins, starch, cellulose. All these are the examples of the imbibants. And lignin and suberin, these two are hydrophobic in nature. That means they do not like the water. Hence, they do not show any imbibition. Lignin and suberin do not show any imbibition. The next one is diffusion pressure deficit. The term diffusion pressure deficit was coined by Mayer. Diffusion pressure deficit is coined by Mayer, and this diffusion pressure deficit is also called as suction pressure. It is given by Renner. The term suction pressure was given by Renner. Diffusion pressure deficit was given by Mayer. What is the diffusion pressure? Each liquid has a specific diffusion pressure. When compared to the solution, pure water has a maximum diffusion pressure and solution have less diffusion pressure. Now, if we see diffusion pressure deficit, what is the diffusion pressure deficit means? At the same atmospheric temperature, diffusion pressure of the solution and the differences between the diffusion pressure of the solution and diffusion pressure of the solvent is called as diffusion pressure deficit. What is this? At the same atmospheric pressure, the differences between the diffusion pressure of a solution and a diffusion pressure of a solvent, that is called as a diffusion pressure deficit. To remove this deficit, solutions, they absorb more water, more solvent molecules, right? That means water always moves from the lower diffusion pressure to the higher diffusion pressure deficit. In this way, diffusion pressure deficit is the absorption capacity of a solution is called as diffusion pressure deficit. And diffusion pressure deficit is also called as suction pressure. Its value is always positive for a cell. And if you see the equation, diffusion pressure deficit is equal to the osmotic pressure minus turgor pressure. Diffusion pressure deficit is equal to the osmotic pressure minus turgor pressure. What it means? When a cell is in flaccid condition, flaccid means what? Normal. In that condition, diffusion pressure deficit is equal to the osmotic pressure. But the entry of water into the cell, that causes the increase in the turgor pressure. That means The increased turgor pressure is equal to the decreased osmotic pressure. The entry of water into the cell will be stopped. That means the cell was in turgid condition. In that condition, OP minus TP is equal to 0. Then finally DPD is also 0. This is called as diffusion pressure deficit.